All right, let's get right into it. Think about this. Inside every living thing, including you, right this second, there's this incredible symphony of chemical reactions going on. It's the basic machinery that powers life itself. And today, we're going to pull back the curtain and see exactly how it all works. So, here's the plan. We'll kick things off with the big picture, what we're calling life's chemical engine. Then we'll meet the tiny catalysts that are really running the show. After that, we'll look at the lock and key model to see how they work, find out what happens when they get put under pressure, and finally, check out where these amazing enzymes are at work in our everyday lives. So, first up, life's chemical engine. This is all about that constant, non-stop chemical activity, this incredible balancing act that basically defines what it means to be alive.
It's a huge question, right? But the answer is actually a single, really powerful concept. It's the sum of every single process that builds you up, breaks things down, and generates the energy you need for everything.
And that concept is metabolism. Simple as that. It's all the chemical reactions happening inside an organism. You can kind of think of it like your body's own internal economy. It takes the food you eat, converts it into energy, and then uses that energy to build everything from muscle to DNA. And this whole economy is split into two opposite but totally vital processes. So, you've got two sides to this coin. On one side, there's catabolism. This is the breaking down phase. It takes complex stuff, like the sugar from your lunch, and snaps it into simpler pieces, which releases energy. Respiration is the classic example. But on the other side, you have anabolism, the building up phase. This takes simple molecules and uses energy to construct more complex ones, kind of like how a plant uses sunlight to build sugars during photosynthesis. It's this perfect dance of demolition and construction, happening all the time. But how do all these reactions actually happen fast enough to keep us alive? That brings us to our next part. It's time to meet the tiny, secret workers that make it all possible. And these guys really are the unsung heroes of biology. I mean, without them, the chemical reactions in your body would be so ridiculously slow that life as we know it, it just couldn't exist. We're talking about enzymes. So what exactly is an enzyme? Simply put, it's a biological catalyst. It's almost always a protein, and it has one supercritical job: to speed up biochemical reactions. And here's the magic trick. It does this over and over and over again without ever getting used up in the process.
And their efficiency is just mind-blowing. First off, they're highly specific. The enzyme that breaks down milk sugar won't even look at anything else. They act incredibly fast. I mean, they can speed up reactions millions of times. They're totally reusable. Their complex 3D protein shape is absolutely essential for their job. And get this, you only need a tiny amount of them to get a massive amount of work done.
Okay, so we know they're super fast and picky, but how do they actually do it? Well, this brings us to the famous lock and key model. To get this, you first have to understand something called activation energy. Picture this. You have to push a huge boulder up a small hill before it can roll down the other side. That initial push, that's activation energy. It's why we cook our food. The heat provides that push to get the reaction started. Every single chemical reaction needs it. And this is the enzyme's genius move. They don't add more energy. Instead, they just lower that hill. They make the push needed to get the reaction started dramatically smaller. It's like they build a tunnel through the hill instead of making you push the boulder over the top. So here's how that unfolds, step by step. First, a specific molecule called the substrate, that's our key, floats over to the enzyme. It fits perfectly into a special spot on the enzyme called the active site, that's our lock. They click together, forming this temporary enzyme-substrate complex. Right then and there, the reaction happens, and the key is transformed into new things, the products. Finally, the products pop out, and the enzyme, the lock, is totally unchanged, ready for the very next key to come along. But as amazing as these little molecular machines are, they aren't invincible. They have to work under very specific conditions. So let's take a look at what happens when our enzymes get put under a little, or a lot, of pressure. Let's start with temperature. At low temps, enzymes are just sluggish. Their activity is way down. As you warm them up, they get faster and faster until they hit their absolute peak performance. That's called the optimum temperature. For us humans, that's right around 37 degrees Celsius, or normal body temperature. But if you keep cranking up the heat, activity doesn't just slow down, it plummets. This is exactly why a high fever can be so dangerous. Because with too much heat, it's not a temporary slowdown. It's permanent, irreversible damage. The lock itself literally breaks.
And there's a word for this: denaturation. The best way to think about it is frying an egg. The heat denatures the proteins in the egg white, changing them from a clear liquid to a solid white. And no matter how much you cool it down, it's never going back to being a liquid. It's the same idea. Extreme heat, or pH, snaps the delicate bonds, holding an enzyme in its perfect shape. The active site gets warped, the lock is broken, and that enzyme is useless forever.
And pH is just as important. It really shows how specialized these things are. I mean, look at pepsin. It lives in your stomach and loves a super acidic environment, around pH 2. But the amylase in your saliva, it needs a pH that's almost perfectly neutral. And then there's trypsin in your small intestine, which needs an alkaline environment to work. It's just a perfect example of form-fitting function, depending on where the enzyme lives. All right, so we've seen what they are, how they work, and what can break them. Let's wrap this up by looking at how we've actually managed to put these tiny biological engines to work in our day-to-day lives. Seriously, enzymes are everywhere once you start looking. You know those bio laundry detergents that are amazing at getting out grass or food stands? They're packed with enzymes like protease and lipase. The lactose-free milk in your fridge, that's made using the lactase enzyme. They use pectinase to make our fruit juices nice and clear instead of cloudy. They're even crucial in medicine, helping people with digestive problems and in big industrial processes like tanning leather. And science is taking this even further. We can now do something called immobilizing enzymes, which basically means we can attach them to a stable material. This makes them way more resistant to changes in temperature or pH, and it means we can easily collect them and use them again, which is a massive game changer for industrial work.
So, it's pretty clear that enzymes aren't just some abstract thing from a biology textbook. They are the literal engines of life, both inside our bodies and out in the world we've built. And as we get better and better at understanding and using these microscopic powerhouses, you just have to wonder, what huge problems are we going to solve with them next?